Hello everyone, this is our interview of what we are reading, and today we are with Clara Peña from the Instituto Amazónico de Investigaciones Científicas (CINCI) in Colombia. Hello Clara.
Hello everybody.
So Clara, you recently published an article in mycorrhizal that is called Distinct Arbuscular Mycorrhizal Fungal Communities Associated with Different Manioc Land Races and Amazonian Soils. So can you tell us a little bit about this publication?
Well, this is, I consider it is a nice publication, an interesting one, because manioc or cassava is a crop species very important in tropical soils. Recently, people have been trying to find a good arbuscular mycorrhizal fungi symbiont for this species. And well, my publication help or make some points about how to select or how to look for that symbiont for this specific crop.
Okay, so one of the main results of the study, for example, is that you showed that the soil type is very important and actually much more important than the variety of the manioc crop. So what do you think are the implications for agriculture in this case?
Well, in the paper, I explained that manioc has many varieties. And the two main groups of those varieties are those that are very toxic, have roots with a lot of cyanohenic compounds, and others that are not toxic. So I suppose that maybe the varieties of cassava will affect in some way the symbiosis, the arbuscular mycorrhizae fungal symbiosis. But I found actually not. It's not very important, the variation between among different varieties. That is more important, the composition of the communities of arbuscular mycorrhizae fungi in the soils.
Okay, so actually the soil component is very important. So that could be something that farmers could work on, like to try to, I don't know, enhance the nutrient availability in the soil, or have you seen something like this?
Yeah, well, I worked with very fertile soils and very poor soils. And what I find is that actually the composition of arbuscular mycorrhizae communities are different, but are not so different. I mean, are different in composition, but not in the abundance or the number of arbuscular mycorrhizae fungal species. That means that actually, even if soils are very different in the physical-chemical composition, you will find there many arbuscular mycorrhizae fungi that can associate with the manioc.
Okay, but there are going to be different communities in different type of soils.
Exactly.
Okay. And so something that also brought my attention when I was reading the paper is that you actually found that there were more diversity of arbuscular fungi in the roots than in the soil, which I find is a little bit surprising because you would imagine that there is a lot of diversity in the soil and only some of them will colonize the roots. So how could you explain these results?
Yeah, well, this has been also found in other papers, and the explanation that people try to do to this issue is that actually because the arbuscular mycorrhizae fungi are endosymbionts, they need to be in the roots, so they will be concentrated in the roots of the plant. In the soil, because the soil is a huge matrix, they will be like diluted. So if you only take a small amount of soil, that is what you need to process it by molecular tools, you will find less fungi there than in a root in which they are concentrated. That is like the response to that.
Okay, yeah, that makes a lot of sense. And so you also discussing a little bit about the methods in your paper, and you showed that actually studies using spores in the soil might not actually be a good representation of the AM fungi that are colonizing the plants. So I think this is very important because, well, a lot of people are using spores to do that kind of study. So what would be your recommendation to researchers that would want to do that kind of research?
Yeah, well, yes, definitely. You have to work with the communities that are into the roots because, as you say, sometimes what you are finding in the soils through the spores are not the same arbuscular mycorrhizal fungi that are colonizing the cassava roots. So maybe yes, but in some cases, and I show in the paper some very nice figures in which you see that actually in some cases are completely different what you are seeing in the root and comparing with the soil. So my recommendation is that actually as people are trying to find a good symbiont or some species that are good symbiont for cassava, they need to, well, use molecular tools. That is the only approach in which you can actually see the communities into the root of the plant. It will be more precise, I will say.
Okay, okay. Thank you very much, Clara. So before we leave you, can you tell us a little bit about what you're doing now? Are you still working with manioc, or do you have other projects that you would like to share with us?
Wow, yeah, well, yes, for me, cassava is one of the main crops and very important for climate change in the future and that kind of things. But we are also trying to use arbuscular mycorrhizal fungi in restoration of degraded lands in the Amazon region. And I would like also to check, for example, how are the communities of arbuscular mycorrhizal fungi in areas in which you have mining. I think and it will be contaminated, for example, with mercury. Mercury. So I would like to check also. Well, those are like future projects, but for now I'm just concentrated a little bit more in the cassava, and we're just pushing the cassava and promoting the cassava as a great crop, and the association of cassava and manioc, that I think now we have more clear things about that association.
Yes. Thank you very much, Clara, for your time. And
Okay.
we will be back soon with more interview for the South American Mycorrhizal Research Network. Bye, Clara.
Bye.